Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my session. So today we are in almost, you can say, the last chapter. So here we are going to talk about the regulation of gene expression. Sorry, not the last chapter, but the last lecture of this chapter. So we are going to talk about regulation of gene expression. So this is the last part of this chapter. So we have completed till translation. So today I'll give you a recap of translation, and then we'll move ahead and talk about the regulation of gene expression. And if time permits, we'll talk about Human Genome Project and DNA fingerprinting. All right. So this is about the last topic. So all those who are watching my class right now, thank you so much for watching my class. Please do like, share and subscribe. So let your friends know here ma'am is teaching the entire biology, both class 11 and class 12. So let your friends know and please bring them as well. Okay, so shall we move forward and talk about regulation of gene expression? So today we'll be focusing mainly on Operon. So what is Operon and why it is present and how it functions? So that is just a small example in the ocean of gene expression. So let's talk about it. All right? Yes? Okay. So here we move forward. So before we go into regulation of gene expression, so today once again we are meeting at 6 p.m. and we are discussing the previous year question. So we started off with your 2019 paper, but we have not yet completed. So today we'll complete 2019 paper and then we'll move forward with 2018 paper. All right? Yes. So please don't forget to join my lecture today. So this is at 6 p.m. So this is at 6 p.m. So please do join. So we will solve some interesting question. Yes, Dharini, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay, so really sorry, yesterday's Menti had to be cancelled because there is a Menti series going on in the JEE channel. So Menti can be opened only at a particular time, only at one site. So we cannot have two studios where the Menti can open. That's not possible. So we had to call off the Menti. So don't worry. So henceforth we'll have the Menti at 4:30 and lectures at 6 p.m. So as long as the Menti series is getting completed. Okay. So today mostly we'll be discussing class 11 topics. All right. Yes. Fine. So come on, Dharni. So please do like, share and subscribe. So don't forget to do that. So this is the PYQ series, okay? And I hope you people have thought about your crash course, whether to join or not. So quickly, the time is ticking off. So the offer price is 1,999 and the original price is 4,500. So make sure you join, okay? So it includes all the 3 subjects, so PCB, and with every day you have each lecture for 1 hour, and every Saturday you have doubt counters, and Sunday you have unit test. So make sure you join me for the same. All right? Okay. So here we move with the topic molecular basis of inheritance. So we were done with replication, then we went forward with transcription. So how transcription happens in eukaryotes and how it happens in prokaryotes. Yes, Antonia B, a very happy good evening. Thank you for joining my lecture. I'm really sorry, yesterday's Menti had to be called off because of the Menti in the counterpart channel, that is eCareerPoint-JEE. So we couldn't have the Menti. But it's okay. So henceforth for the next 10 days till their Menti series gets over. So if we are having a Menti, so Menti will be at 4:30 and lecture will be at 6. All right? Yes? Okay. So here we go. So we spoke about the genetic code. So we also spoke about mutation and genetic code. So we also have TRNA. So we described in detail about TRNA, which is the adapter molecule. And then we started talking about translation. Yes, and I also finished about translation, but giving you a quick recap of how it happens. So the Menti will happen on Monday. I will let you know tomorrow. All right, Anthony? Yes? So Anakin, hello, hi. So I guess you're new to my channel. So hello, thank you for joining my channel. Thank you for joining my lecture today. So please do like, share and subscribe. Okay, so today we're going to talk about regulation of gene expression. So before we go into that, I want to give you a recap of translation. Okay, so how translation happens. So translation is when the information from the mRNA has to be decoded to a polymer of amino acids, right? So this is your proteins, right? So here this is 5 prime, this is 3 prime. So here I have poly A tail, and here I have guanylyl methyl triphosphate cap, and here I have exons, right? So this is how it has to be translated from here to here. But how? So you have 4 nitrogenous bases here. So since it is mRNA, so you can say 4 nucleotides instead of nitrogenous bases. So I will change it. So it has 4 nucleotides. Okay. A, G, U and C. Since it is mRNA, so you have U here, right? So these are the 4 nucleotides, and in proteins you have 20 amino acids. So how does it code for? So that is when we came to the discovery of your genetic code, that is 4 power 3 is 64, right? So which means each of the message is in the form of 3-letter codon, and it codes for 64 amino acids. So I hope you remember how to do your genetic code fully. Yes. So I hope you remember how to do the crossing. Fine. So your genetic code, though it has 64 codons, only 61 codes for your amino acid. Okay, and the remaining 3 codes for your stop codon. So what are the stop codons? Do you remember them? So it is UAA, UAG and UGA. So these are the stop codons, alright? Yes? Okay. So what are the features of your genetic code? So number 1, you have 64 codons. So this is the first part. So out of which 61 codes for amino acid, right? And we have 3 codons which are stop codons, right? So this is the first part. And then you have many codons which codes for a particular amino acid. So you have many codons which code for one amino acid, right? So this condition is known as degenerate. And your genetic code is read in a continuous fashion. There is no punctuation, etc. So your genetic code is read in a contagious fashion. And number 4, it is universal, right? So genetic code is common back from bacteria to eukaryotes. So everywhere U codes for phenylalanine, and everywhere C codes for proline. Alright? Okay. So then the last one is that you have an initiator codon. So this initiator codon is AUG and it codes for methionine, right? So these are the features of your genetic code. Yes, you're right. So now, how does the process of translation begin? So the translation includes 3 steps. So what are the steps of translation? So one is initiation. The other one is elongation, and then you have termination. Right? So these are the 3 steps of your translation process. Methionine are cysteine and sulfur containing methionine and cysteine are sulfur containing amino acids. Yes, Antonia B, you're absolutely right. Okay. So this part is done. But before we go into the process of translation, if I have an mRNA here, this is 5 prime to 3 prime. So who is going to read the codon and translate into proteins? So who is going to be the translator here? So here, thus the adapter molecule is the tRNA. So T stands for transfer RNA. So initially it was also known as soluble RNA. Fine. So we spoke about the structure of your tRNA. So tRNA structure is like this. Right. So this is the structure of your tRNA. So this is your amino acid acceptor end. So this is your TC arm. So thymine pseudouridine arm. So this is your D arm. And this is your anticodon arm. So this is going to read the message from your mRNA and then convert it further. Alright, so this is your adapter molecule. So now, if you have the information on mRNA, your tRNA should come and bind. Yes, Shimpi, hello, hi, a very happy good evening. I'm good, thank you. How are you? So Shimpi, so please do like, share and subscribe. So Shimpi, how are you now? You're completely fine? Yes, no cold. So please don't fall sick. So one more thing here you have to remember. So the process of translation happens in your cytoplasm. Right. So now your tRNA carries a particular amino acid. So tRNAs are specific for amino acids and there are no tRNAs for your stop codon. Right? Okay. So first your amino acids have to bind to the tRNA. So this is the first step. So the first step of initiation is that the amino acid should bind to the tRNA. So this happens in the presence of ATP molecule. Okay, and it also happens in the presence of an enzyme known as amino-acyl tRNA synthesis. So this step, which initiates the process of translation, is also known as charging of your tRNA. So charging of tRNA happens in the cytoplasm and not on the ribosomes. So first an amino acid should attach to the tRNA, to the specific tRNA. For that it requires ATP and it also requires amino-acyl tRNA. So this step is known as charging of tRNA and it happens in your cytoplasm. Alright? Yes? Okay, so now what happens after your tRNA is charged? Now it will come and bind to the ribosome. Right? So this is the largest subunit. This is the smaller subunit. So I have my mRNA like this. So my tRNA will come here. Yes, it is clover leaf shape. Yes, you're right. So whoever told me, yes, okay. So it comes in like this. So your largest subunit binds to the, your tRNA enters the largest subunit and the TC arm helps in binding to the larger subunit. Okay, so it is like this. So if I have my mRNA, if on mRNA, if I have a coding sequence like this, so this is 5' to 3'. So this is the first base, second base and third base. But on my mRNA, on my tRNA, so this is U, A, C. So this is first, second and third. Because in this order it binds, right? So please remember this. Yes, hello, hi. Munna, hi. Yes, I'm good, thank you. How are you? So Munna, please do like, share and subscribe. Thank you for joining my lecture. Yes, Anakin, you're right. It is clover leaf-like structure. So Havila, hello, hi. So thank you so much for joining my lecture today. So please do like, share and subscribe. Hi Keshav, what a surprise, you're here. I hope you're doing good. So thank you so much for joining my lecture. So please do like, share and subscribe. Alright, yes, so this is how it is. So here giving you a quick recap of what the genetic code is. right? So he was a physicist, his name was George Gamow. So he suggested that you have 4 bases and 20 amino acids. So the codon should constitute a combination of bases. So he proposed that it is 4 power 3. So what is the name of the scientist? George Gamow. Okay. So to prove that this is right, 4 power 3, 64 we found out, and we also found out each codon is a 3-letter pair, right? But how did they actually find this out? Yes, Avinash, so hello, I guess you're new to my lecture, so thank you so much for joining. So please do like, share and subscribe. Oh, Kal, you got fever. Why, Munna? Please take care. You got fever from Shimpi. So please take care, Munna. That's why I know you're not well, so that's why I cancelled the mentee. Okay? Yes, Swami. Hello, hi. A very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Hi, Guha. Yes, welcome. A very happy good evening. Thank you so much for joining my lecture. So Keshav, what happened? Why this red face? Because it's very hot in your place? Yes, it's really hot in Kota as well. Okay, so who did actually the proof for this? Yes, Shivam sir, hello, good evening. Okay, so the chemical method, the proof was done by Hargobind Khurana, synthesizing an RNA molecule with defined combination of bases having a homopolymers and copolymers. So someone has to prove that this is how it actually happens in the cell, right? Yes. Okay, you are feeling bad for Hunda. Okay, Keshav, right? Okay. Yes, guys, so please take care. So please don't fall sick. So it is not advised to fall sick. So please make sure you don't even have any smallest of the illness as well, because your exams are nearing. So we don't know when CBSE will announce when NTA will announce the date. So please stay safe. Okay. So Munna, so please stay safe and ask Shimpi also to stay safe. Okay. Yes. Fine. So, if this is the condition, we have to prove this is how it happens inside the cell. We don't want any stories, we actually want to know how it happens inside the cell, right? So, Hargobind Khurana, so he was an Indian, so I told you even today we have scholarship in his name for all the Indian students. So, he artificially produced a mRNA molecule. So which means the mRNA had bases as well. So we had to artificially synthesize nucleotides and then make up your mRNA. So he helped in the proofreading of this concept. Then you have Marshall-Nirenberg's cell-free system where protein synthesis helped to code the deciphered. So that is Marshall. So all this process of central dogma happens inside your cell. But to cross-check this, they did it outside the cell, right? It was a cell-free system. So directly they took an mRNA and they wanted to check using this code that yes, it codes for a protein. So these two scientists helped in proofreading. So why am I talking about this? So these two scientists' names are mentioned in your NCERT. So you should remember. Alright, the third scientist is Severo-Oka enzyme. Okay, so he was the person who discovered it. So polynucleotide phosphorylase. So this was the person, scientist Oka. So he discovered the enzyme polynucleotide phosphorylase. So it is known as PPP. Okay. So it's helpful in polymerizing RNA-defined sequences in a template-dependent manner. Suppose to find an RNA to make a synthetic RNA is tough. Okay, so if I make a DNA and then I convert it into mRNA, it is a bit easy. So all these people help to prove that this is how your translation process happens. Alright, so charging of tRNA, so I told you amino acid combines with ATP, then you have amino acid AMP. ATP is getting used, so it's amino acid AMP, and now it binds to a specific tRNA. So now this tRNA is known as charged tRNA, right? So this is the structure of your tRNA. So you have your amino acid arm. So please remember, amino acid arm is present at the 3' end of the tRNA. So amino acid acceptor arm is at the 3' end. Okay, so this question is frequently confused, so please remember this, and this is known as TC arm. So this symbol is known as pseudo. here. So it is thymine pseudouridine arm. It helps in attachment to the larger subunit of your ribosome, and this is your D arm, dihydrouridine arm. Okay, so this stands for dihydrouridine arm. Alright, and this is your anticodon arm. So what is wobble hypothesis? So if you understand, okay, so this is how it happens. So your tRNA, so they bring the amino acid, they help in the formation of peptide bond, and they move away, right? I told you this is E site, exit site, the peptide site and the A site. They come inside and they have the particular amino acid, then they move forming the polypeptide and then they exit, right? Okay, so here what is wobble hypothesis? So before that, the process of polymerization of amino acids to form a polypeptide is your translation. So the first phase is your tRNA gets activated, then you have the peptide bond formation, etc. So this is the GIF I told you. Yes, so this is how it happens. So I hope you remember. So this is the exit site, E site. So peptide forming site, P site, and this is the site of attachment, that is A site. All right. Okay. So now you should remember what is wobble hypothesis. Suppose if I take arginine. Okay, so all these codes for arginine, okay, but if you watch closely, the first two bases are common, right? Yes, only the last base changes. So the last base is prone to susceptibility. The moment if you have your mRNA like this, 5 prime to 3 prime. So if I have a sequence CGU, first base, second base, third base. So in the corresponding tRNA anticodon arm, so here I have G, C and A, right? So this is the third base, second base and the first base, right? So depending on the first base, it can, so depending on the third base of the mRNA and the first base of the anticodon, it can wobble, it can switch between amino acids, okay? But for all the three, it needs the same tRNA. For each amino acid, you have a tRNA, right? Since all of these codes for arginine, it has the same tRNA. So the third base determines the specificity. So this is known as wobble hypothesis. So nearly you have 31 tRNAs plus 1 initiator tRNA. So totally we have 32 tRNAs. So first two is the same, right? So what defines which amino acid it will code depends on the third one. But for all of them, we have the same tRNA. So this is your wobbling hypothesis, okay? Coming to gene regulation. So with that, we end translation. Now we move forward with your gene regulation. So what is gene regulation? So anything and everything has to be regulated, otherwise it ends in a chaos, it ends in a problem, right? So everything has to be regulated. So if YouTube is not properly regulated, then you have n number of videos which are bad videos will be added. So there is no control. So you as a student, you should be controlled. You should be only given good content, you should be allowed to have good food, you should not be given wrong food, you should not be given drugs. So everywhere there is regulation, right? So the point of regulation starts from a gene. So our DNA is 2.2 meters and in that we have 25,000 genes. So each gene codes for a particular protein or a product, right? So we have 25,000 genes, which means we are going to have n number of products. So at a single time, if you are going to have all the 25,000 genes operated, your cell will burst. That is not possible. what when needed will be present at a time. For example, now you go to the grocery store, you get all your groceries for the next 1 month. So we do not shop groceries for the next 10 years, do you? 9. So you don't even shop groceries for the month of November. You make sure you have enough groceries for the entire month of July, and you make sure you have groceries for the first week of August, right? So maximum only that much you can shop. So similarly, whichever protein is needed right now, only those will be expressed right now in your cell. All the proteins will not be expressed. So that is known as gene regulation. Yes, Ayappan Shakti, well, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Alright, so this is one way. So the fundamental problem of chemical physiology and of embryology is to understand why tissue cells do not all express all the time all the potentialities inherent in the genome. So this was discovered way before. So these lines were said by Francis Jacob and Jacques Monod, who were important people in your enzymes, okay? So out of the 4,000 genes in the bacterial genome, 35,000 genes in the human genome, only a fraction of them is expressed in a cell at any given time. So this is the rule of your cell. Yes, hi Satyendra, hello, very happy good evening. Yes, I'm good, how are you? So beach mein you got disappeared, so how are you, where are you? Are you fine, you're doing good? Yes, have you come back to Kota kya? Huh? Okay, so only a fraction of them is expressed at a given time. So some gene products are present in very large amounts, whereas some are present in very small amounts. For example, EFs are elongation factors. So elongation factors are needed in protein synthesis. So this can be translation or transcription. So both of them are needed for protein synthesis, right? So these are present in excess amount in bacteria. So ribulose 1,5-bisphosphate carboxylase oxygenase, which is your Rubisco, it is also present in abundant content, okay? So where can it regulate? Suppose, for example, if you order a particular parcel in Amazon, right? So you order because you think you need it, okay? So you order a raincoat, so you need a raincoat because it's rainy season, so you're ordering the raincoat, okay? But suddenly you find out that you have to move to a hotter region. Say for example, you are in Shimla where it's raining and you suddenly have a situation where you have to move to Kota. So you would cancel the raincoat, right? So you know how to cancel, and there is this option. So similarly, if a cell thinks it needs a particular gene, particular protein, it will continue. But suddenly it can get an environmental signal which says it is no more needed. Then the cell has to abort that process. So these are levels of regulation also, okay? Yes? Alright, okay Satyendra, fine. So please take care. So this is levels of regulation. So cellular concentration of a protein is determined by a delicate balance of at least 7 processes, each potential points of regulation. So first, where I can regulate my synthesis is the synthesis of the primary RNA transcript. So right from my DNA strand, so 3' to 5' is my template strand. Right? So when RNA polymerase does this job, okay, so I have a primary transcript. So if the cell understands it no longer needs just product of this gene, it can delete or degrade this primary transcript. So this is level 1. Then post-transcriptional modification, where you have capping and tailing. Even after it is capped and tailed, if the cell comes to know that this is no longer needed, it can undergo the process of degradation, okay? The next one is mRNA degradation. Even after the mRNA reaches the cytoplasm for translation, the cell has the power to regulate its gene. Then protein synthesis, when the process of translation is beginning, it can control. Once translation is over, the proteins have to be packed to whichever region it has to move. So in that packaging also the cell can stop its process And finally, when the protein has to move, what is transcription? Okay, so transcription is the process of copying the information from DNA to mRNA. So this is known as transcription. So from DNA you copy to mRNA, so this is transcription. And from mRNA, when you produce proteins, it is translation. Okay, Avinash? Yes? Okay? Yes, that I know. Yes, I understood. So protein targeting and transport. Now, for example, the protein produced in the cytoplasm has to reach the hypothalamic cells or has to reach the nephrons. So there also it can be stopped. Finally, the protein can be degraded. So these are the 7 levels of regulation. So from the DNA, so on the DNA you have gene. So the gene is getting transcribed to an mRNA. So here also you can delete the mRNA where your cell can do, and then you have post-transcriptional processing. The cell can inhibit this process so that the mRNA degrades. Okay, even after an mature mRNA is formed, it can still do degradation. Fine. Now the mRNA has moved to the cytoplasm. Now this is translation. So here also it can stop the process or halt the process. Then it is post-translational processing. Yes, for packaging to a particular place, and during transport also it can hinder, and finally it can degrade the protein itself. Even after the final process is done, even after you receive a raincoat from Amazon, you have the option of return, right? So these are the levels of regulation. Okay, so to have regulation, you need some criteria. To return a parcel in Amazon, you have to ensure the product is not damaged. You have the bill, so you should not dirty the product, right? So there are certain terms and conditions. So similarly, for principles for the gene to be regulated, you have certain principles. So you have what is known as housekeeping genes. So in an office, it's okay if a particular, so see, in a school, it's okay if a PT teacher does not come for a month. It's okay if a science teacher does not come for a month, but it's not okay if the cleaners do not come for a month, right? So the place will stink so badly. So housekeeping has to be done properly every day. So imagine the maid in your house does not come. So yes, 1 day, 2 days you can do, but how many days will you do, right? For those who have maid. Suppose if your mom has gone for a vacation, your mom has gone to some place and you have to manage the house. How bad you feel, because you have to manage everything. Yes, so you need someone who can maintain the house, you need someone who can keep the regular activities in check, right? So in your cell also you have something known as housekeeping genes. So these genes are continuously transcribed and translated because they are needed always, okay? So genes expressed at more or less constant level in virtually every cell or a species or an organism. So that is your housekeeping gene. At all time it is present in the same level in your cell because it is needed at that level. It is not in high amount or low amount, it is in the constant level. So this is your housekeeping gene. Offline classes, no, offline classes are not open till September because now they say the second wave is about to come, right? No, offline classes are not yet open, Satyendra, okay? So the unvarying expression of the gene. So there are certain genes which are known as housekeeping genes whose level remains the same. They are always needed by the cell, so we call it as housekeeping genes. But their levels do not change, right? They are always present at a constant expression. So they are known as constitutive genes. So they are known as constitutive genes, right? So housekeeping genes have a constant expression. So they are known as constitutive genes. So the gene products which in the cellular levels rise and fall in response to a molecular signal. Suppose if I have a signal from hypothalamus. So if I have gonadotropin releasing hormone, So then my pituitary gland will produce gonadotropins, that is FSH and LHs, right? So depending on a particular signal, these are getting transcribed, right? So these are known as inducible genes, right? So depending upon a signal, so these are getting activated. So these are known as inducible genes, and the process is known as induction. Fine. So similarly, you have feedback mechanism. Like for example, the final hormone itself will stop the function, right? So you have inhibin, which is secreted by your ovary, which will stop the synthesis of your gonadotropin. So depending upon an environmental signal, so this concentration is reducing. So this is known as repressible gene. So depending on an environmental condition, if the process slows down or if the concentration slows down, it is environmental process, it is known as repressible, and the condition is known as repression. So induction is when you induce something, the concentration increases, that is induction. When you act on something and the concentration decreases, so that is repression, and the genes are known as repressible genes. Alright, yes? No, Satyendra, we are not taking any offline class. So day in and day out, it is online class only. Okay? Yes. So now, what is an operon? So I told you in gene expression, you have 7 levels of expression, and you have to remember what are housekeeping genes, what are constitutive genes, what is induction, and what is repression, right? So now let's talk about what is an operon. So the gene cluster and promoter plus additional sequences that function together in regulation is called an operon. So can you see? All these are coding sequences. So all these are associated with a single promoter, single operator, and single regulatory sequence. So this is known as an operon. So when you have many cluster of gene, so this is a gene, this is a gene, and this is a gene. So when you have a cluster of genes, okay, attached to a single promoter and additional regulatory sequence, it is known as an operon. Alright. So what is constitutive gene? Okay. So there are some genes in your cell which are always present. So those genes are called as housekeeping genes. So these housekeeping genes have a constant level of expression. They are always expressed in the same level. So this constant level of expression is known as constitutive gene expression. Okay, Avinash, is this clear? This is known as constitutive gene expression. They are continuously expressed, and they are also expressed in a constant level. Okay, yes ma'am, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Okay, is this clear? So operon is something which has a structural gene, has the same promoter and same regulatory sequence. Okay, so an operon can include 2 to 6 genes transcribed as a single unit. I have only one promoter. If RNA polymerase binds here, so all of them will be transcribed at once, right? So you have different types of operon. So you have lac-operon. So lac stands for lactose. You have TRP, this is tryptophan operon. So you have ARA, this is arabidinose. So H is for histidine and val is for valine operon. But your textbook discusses only lac operon. So we will also talk about your lac operon now. So what is lac? So lac stands for your lactose, right? So what is lactose? So lactose is a disaccharide. It is a type of sugar, right? So lactose can be broken down into glucose plus glucose. So these are individual monosaccharides, right? Suppose in a bacterial cell, if glucose is very less and the media or the food that it receives is only lactose, the bacteria should be able to digest or metabolize this lactose. right? Yes. So for that we have a lactose operon. Okay, so it has a cluster of genes. So this is your structural gene, right, which codes for a particular product. So these are your structural genes. Fine. So gene Z codes for beta galactosidase. So beta galactosidase helps in the conversion of lactose to glucose plus glucose. So here is where beta galactosidase functions. Fine? Yes. Now gene Y codes for an enzyme known as permease. So permease permits the entry of lactose inside the cell. So permease permits lactose inside the cell. Okay. The last one is gene A. So gene A codes for trans-acetylase enzyme. So this enzyme helps in the transfer of acetyl group. Okay, so these are the 3 protein coding genes. Now, this P stands for a promoter. So, promoter is the one which initiates any process, right? So, this promoter is for this inducer. So, I here stands for inducer. So, this promoter is for this inducer. I have another promoter, so this is also promoter, but this promoter is for all these genes and this is my operator. Yes, okay, right, fine. So now what happens? If lactose is present in the bacterial cell, I need all these genes, right? It makes sense? If lactose is present in the bacterial cell, I need all these 3 genes, right? So then I need to have this transcribed and translated because it has a purpose. But if there is no lactose in my bacterial cell, if there is no lactose as food for the bacteria, if it is directly glucose, then what is the use of these genes? There is no use, right? Yes. So now the cell can stop its transcription and translation. How? So if lactose is absent, so this is case 1 when lactose is absent. So when lactose is absent, we do not need all these enzymes. So when lactose is absent, the inducer produces a repressor mRNA. So this is your DNA, right? So the inducer promotes it transcribes to form a repressor mRNA. So this is the promoter, so this will initiate the inducer to produce the repressor mRNA. So the mRNA produces a repressor protein. So this repressor protein will go and bind to the operator region. So when this repressor protein goes and binds to the operator, the RNA polymerase cannot move further. It cannot cross this place. Okay, so these genes are not getting transcribed. So when lactose is absent, so these genes are not getting transcribed. Okay, how? With the help of your repressor protein which binds to the operator region. Okay, yes. 1 to 7 hours. Okay, fine. All right. So let me try. Fine. Okay. So this is when lactose is absent. So when lactose is present, So once again, the promoter, so RNA polymerase binds to the promoter, the inducer produces a repressor mRNA and it produces a repressor protein. But since you have lactose, the lactose goes and binds to the repressor protein. So now your operator region is free, so your RNA polymerase transcribes this region. So this is when lactose is present. Can you see, depending upon the need, the cell is taking its function forward. Yes, done. So Satyendra is asking me to have a marathon class for the entire class 11th biology in a single day for 6 to 7 hours. Oh my god, that itself is really shocking. So Monday to Saturday I have 7 hours or 6.5 hours classes, and Sunday again you want me to take for 7 hours? Huh? But yes, when we near the NEET exam, let me think about it. So think about joining the crash course, Satyendra. So there is a crash course for 1999. It is completely in English. So it's PCB. The offer is for a very short time. So biology I am taking. So every day you have 3 hours of lecture, 1 of physics, chemistry and bio. Every Saturday you have doubt counter and every Sunday you have unit test. So think about joining this, okay? The course commences from 15th of July in another 6 days. So please think about it, alright? Yes, so is the lac operon clear to everyone? Yes? So with this we are done with the lac operon. So this is how gene regulation happens. Done everyone? So Anthony, is it clear? Shimpi, Dani, Grace, Guha, is it clear to everyone? Yes? Yes, alright. So with this we are done with the lac operon. So here I have tryptophan operon. So this is not there for your NEET exam, but just to show you how it operates, okay? It has 5 genes. So your lac operon had 3 genes. So this has 5 genes. So when your tryptophan is present, so TRP is tryptophan operon. So when your tryptophan repressor, so here R stands for repressor. So once again your repressor produces an mRNA and it produces a repressor protein, okay? So now this repressor protein, okay, if your TRP is present, your tryptophan binds to this repressor protein which goes and binds to your operator region. So then all these regions are not getting transcribed. It is the vice versa here. So in lac operon, when lactose goes and binds, the transcription happens, right? Here in tryptophan operon, if tryptophan goes and binds, so this is to reduce the high level and the low level expression. So when you have tryptophan binding to it, this region is not transcribed. It's okay, don't confuse more. This is just an example. Alright, so we are just left with 2 small portions, that is Human Genome Project and DNA fingerprinting. So which I will cover on Monday, because anyways we have to discuss important questions from this chapter. So all together I will finish it on Monday. Alright, yes. So make sure you join for my 6 p.m. lecture. So 6 p.m. we are discussing about your we are discussing your previous year question papers, right? So all this I will teach you on Monday. So VNTR everything. So yes, so I have my lecture once again at 6 p.m. So please join me. So we are going to discuss 2019. We are going to finish 2019 and we are going to start with 2018 paper. So Satyendra, this is the crash course I was talking about. So please think about joining this. So this is useful for all those who are going to appear for 2021 NEET exam. Okay, yes? Alright, okay Satyendra, that's fine, no offense. So if you genuinely liked my way of teaching, so please do like, share and subscribe, and hold on for an announcement at 6 p.m. about tomorrow's session. I have something interesting for you, alright. So thank you so much for making the lecture very interesting. So I wish to meet you at 6 p.m. as well. So until then, stay tuned and take care. Bye bye.